I have the great honor, but also a large duty to basically give you an overview of where we are in terms of the state of the art in bioelectrochemistry as a sort of subfield and where we're going. So I will give it my best. But we can't really talk about sort of where we are without talking about sort of where we've been. And so I wanted to just introduce, if I can manage to get this to work. There. I wanted to sort of remind everybody that even though I'm talking about sort of things that are happening right now in bioelectrochemistry, we're basically standing on the shoulders of giants, people like Harry Gray, who really taught us how to do protein electrochemistry, Adam Heller, who taught us how to make glucose biosensors, Ralph or Buzz Adams, I didn't meet him ever, but essentially basically showed that there was an interaction between electrochemistry and the ability to actually look at neurons and look at neurochemistry. And then after that, Mark Whiteman, who really made next steps in terms of being able to do single entity, in this case single neuron measurements. I think we're going to hear more about single entity electrochemistry from Jeffrey Dick later in the morning. And then these guys that you may or may not recognize. So all of these people show up to ECS meetings occasionally, and so you've probably seen them at ECS or at ACS. But these guys actually developed patch clamp techniques. And so we learned yesterday at the end of the day that 30 years ago, mass spec, you had to build your own instrument, and you had to be an expert in mass spec in order to do mass spec. And today, mass spec is a commercial technology that all of us can, buy one and do our own mass spec. And so patch clamp is basically, in bioelectrochemistry, that technique that started out as ideas for these two guys who won the Nobel Prize for it, but now is a commercial device that isn't used too much by electrochemists, but instead is actually used by people in the biomedical community, in the biosciences, who are using patch clamp to try to understand how cellular processes work. So we've kind of made full circle where we went from something that you had to be an absolute expert in to devices that you can buy and do your own bioelectrochemistry, even if you're not an electrochemist. So that's kind of where we were. I'm going to talk about different areas of bioelectrochemistry today. And for all intents and purposes, the definition of bioelectrochemistry is that you're either using electrochemistry to look at a biological system, or you're actually wiring a biological system to an electrode and using that electrode for stimulation. And so I'm going to start in the beginning, actually talking about situations where we're using kind of traditional electrochemistry, but in a biological environment like neurochemistry. Then I'll talk about biosensors, because this is probably the part of bioelectrochemistry I think everyone is probably most well versed in. And then we'll talk about all of these other different techniques and different areas of bioelectrochemistry that are becoming popular today. So if we look at where we are in terms of neuroelectrochemistry, we've made a lot of progress in terms of actually being able to do the electrochemistry, and we're using that progress to actually look at how disease progression works. So we have a number of people across the U.S. As well as across the world who are miniaturizing electrodes so that they can actually do either in vivo, so you may be putting these electrodes actually into a rat brain, or single neuron evaluations to be able to understand how things like dopamine release, how you're producing neurotransmitters, and basically how these sort of signaling molecules may actually be playing a role in either disease progression for things like Parkinson's or Alzheimer's, or actually in terms of possibly understanding mechanisms and therefore taking it the next step to thinking about therapeutics. So a portion of that has really been sort of materials design. So how can we make electrodes smaller? How can we make them more durable? How can we make them biocompatible so that we can think about all of those applications? So I'm showing some examples here that are actually used for looking at traumatic brain injury. So these are actually would be implanted sort of in vivo analysis. But we also spend a lot of time in terms of, looking at the fact that you may want to look at a single neuron and how you have release from a single neuron, so sort of single entity detection. So this is some work from Jill Venton's group, where she's actually been making carbon nanofibers that have been modified to be able to have sort of better selectivity and better performance. So as you can see, this is both about making new materials, things like carbon nanotube yarns, as well as thinking about sort of next generation techniques. We're all really comfortable with cyclic voltammetry, but going to fast scan voltammetry to be able to get the sensitivity and the selectivity that they would like to get to be able to do this analysis. So a portion of the bioelectrochemistry community now is working on those materials to be able to essentially look at these in vivo or cellular systems. That's actually not the area that I work in, and so I'll tell you a little bit about the area that I work in that is sort of maybe less biomedical and more focused towards sensing in general, as well as energy and water. The energy-water nexus, so to speak. So in order to do that, I am basically very interested in bioelectric catalysis. So I'm interested in being able to use enzymes or microbes as catalysts. And when we talk about using an enzyme or a microbe as a catalyst, we actually have multiple different routes by which electron transfer can happen. So if we have a system where we basically have a very buried active site, we may have to add mediators to be able to mediate electron transfer from wherever our biocatalyst is to the electrode surface. So from the microbe to the electrode, from the protein to the electrode. Some proteins and some microbes can actually do direct electron transfer. So that means that they actually have active sites that are capable of tunneling electrons from the electrode surface, or from the enzyme to the electrode surface, or the electrode surface to the enzyme or the microbe. And some of them do interdomain transfer, which basically means that you have a lot of redox species in some proteins, and you definitely have a lot of redox species in a microorganism. And so you may actually be doing internal electron transfers as you sort of make a pathway from wherever your active site is to the electrode surface. So for all intensive purposes, a mediated wire, so to speak, where you're mediating those electrons from the active site to the electrode surface. For all intensive purposes, mediated electron transfer is the easiest thing to do. Direct electron transfer, you'll see a lot of controversy in the literature because some proteins can do direct electron transfer, some can't. So if you think about this, you have to be within tunneling distance, and there are some microorganisms that you cannot get sort of surface cytochromes that can tunnel. Some enzymes, you basically have the active site so buried within the enzyme that you can't actually easily do the transfer. So direct electron transfer is usually our holy grail, our goal, but we probably spend a lot of time doing mediated electron transfer. And we see that most frequently when we actually think about sensors. So the most common sensor, electrochemical sensor that you would buy today from a biological perspective is actually a glucose sensor. So if you go to CVS or you go to Walgreens, and you actually buy a test strip for testing blood glucose, that is actually a glucose oxidase-based biosensor, and there's been a series of generations of glucose oxidase biosensors. The first generation, the glucose oxidase, when it oxidized glucose, it would actually take oxygen that was dissolved in the blood and produce peroxide. And actually, the first generation sensors actually looked at the depletion of oxygen. So as this reaction was happening, the oxygen is being depleted. Thank you. The second generation was a little bit similar in that it was actually looking at peroxide production. So again, you're doing the electrochemistry. You're not necessarily doing it at the electrode surface, but you're producing something from the enzymatic. Sorry, I should say, the enzymatic chemistry is not necessarily happening at the electrode surface, but you're producing a product that you can detect at the electrode surface. So your electrochemistry in that case would be peroxide production. Third generation is a mediated scheme, like I just showed you, where we have a mediator that's mediating that glucose oxidation by glucose oxidase. So those are the three types of commercial systems. We're working on fourth generation direct electron transfer systems, but they're not really out in the market yet. So what you typically see in the market is something that looks like this. It's usually for glucose, although there are lactate systems now. People are working on peroxide, pesticides, nerve agents, DNA, bacteria, fungus, viruses, any number of things using this sort of same concept that you essentially have a mediator that is going to mediate the electron transfer from wherever your biocatalyst is. In terms of glucose, the blood glucose monitoring market is quite large and getting larger as we have sort of more diabetic patients, especially. So this is the market just in the U.S. We're seeing an increase in the diabetic population here in the U.S. that is causing an increase in the market for blood glucose monitoring. So these are our biosensors. This same concept of essentially using a biocatalyst for sensing can actually be used for other applications. So we don't just have to use that for analytical applications. As we start to think about designing these, there's a lot of variables that go into this. So choosing your mediator, choosing your enzyme, choosing your polymer that's going to coat it onto the electrode, choosing your cross-linker and how long you're going to cross-link and what the concentrations are. And right now, this is still done kind of by trial and error, where you will make a 96 well plate of different electrochemical sensors and evaluate them. And as you evaluate them, you'll say, okay, I like this particular enzyme, I like this particular mediator, I like this particular polymer because it works well with the enzyme that I chose, but it's all kind of trial and error. And I think if you look at sort of where we're going, and you'll see a bit of this from Matt Sigmund's talk later, is that really you can use some of the machine learning tools, some AI tools, some computational modeling to actually start predicting better mediators. We can use enzyme engineering to actually make better enzymes, and then we can learn about polymers that make good sense in terms of intermolecular interactions. So if we think about sort of translating that concept of kind of wiring a biocatalyst to an electrode surface, we can use it for sensing. We can also use it for power generation. So I'll talk a little bit about biofuel cells, where really the only difference between a biofuel cell and a traditional fuel cell is a traditional fuel cell typically has a platinum or platinum alloy. There are some non-precious metal catalysts, but basically a metal catalyst at the anode and the cathode. And a biological fuel cell replaces either the anode catalyst and or the cathode catalyst with a biological catalyst. So that could be a living organism or that could be a protein, but some type of biological catalyst. And so this is done quite a bit for both microbial systems as well as enzymatic systems. So this is an example from Lenny Tender and Derek Lovely of basically a first generation microbial fuel cell where they basically have a carbon anode and a carbon cathode that are actually placed in this 50-gallon drum that has holes in it, and that is actually placed in the sea. And since the sediment in the ocean, or this could happen in any sort of waterway, has a lot of carbon material in it, we actually can take that sediment organic material and the biological organisms that will actually grow on those carbon electrodes will actually oxidize them. So here you're naturally growing a biofilm. That natural biofilm is going to act as a catalyst. In this case, it's not a design system. That biofilm is actually going to naturally form with time and start to generate power. You're not going to get a great deal of power out of this, but you can power buoys, sensors with this particular technology. So in the early 2000s, they were focusing on this for a lot of military type applications. Today, you see more people looking at microbial fuel cells as a way to do wastewater treatment. And so this is a company in California that is actually building very large reactors to be able to do electrochemical wastewater treatment with a microbial fuel cell. So they're essentially decreasing the energy needs for wastewater treatment by basically having an anode that is going to be coated with a biofilm to be able to oxidize things that are in wastewater, so things that are in the sludge and the wastewater, to be able to generate some energy to sort of decrease the cost of wastewater treatment. So we kind of transitioned from something that kind of had a military application to something that is really focused on improving wastewater. We can do the wastewater treatment. We're also seeing a lot of work where people are taking this the next step to do electrosynthesis with microbes, where they're actually using the microbes to be able to produce chemicals like hydrogen and methane. And with synthetic biology, we can think about actually making microorganisms that will do a much wider variety of chemistry. And I'm going to show you one example from my lab where we basically took the NIF gene, which is basically a nitrogenase gene. So Tom talked yesterday about nitrogen reduction and how difficult it is to do nitrogen reduction to ammonia. But there are in living organisms an enzyme that is capable of doing nitrogen reduction to ammonia. It's called nitrogenase. It works at room temperature and ambient pressure. And so we can actually take that gene from a nitrogen fixation bacteria, and we can actually introduce that gene into other microorganisms that are electroactive to be able to do electrosynthesis of ammonia. So in this case, we basically taken a cyanobacteria, and we have introduced the chemistry so that the cyanobacteria will produce this nitrogenase, produce this protein, so that it can actually produce ammonia electrochemically after the biofilm forms. So we're indeed using synthetic biology and the ability to put non-natural genes into bacteria that can actually communicate with electrodes to actually produce chemicals. Now, if you look at the microbial fuel cell community, you'll see people initially studying things in H cells or U cells. You're starting to see some translation to fuel cell stacks like you would see in a traditional fuel cell. But a lot of the reactors that you're seeing actually look more like the bioreactors that they're actually using for wastewater treatment now, but now they're incorporating electrodes in them and sort of thinking how they can design electrochemical cells to be both the microreactor that we want as well as the electrochemical cell. So sort of a combination. So if you look at this community, you'll definitely see a lot of fuel cells that don't look exactly like the fuel cells that we saw yesterday in the talks. If we don't put entire living organisms down, we can actually put catalytic enzymes, so the portion of the living organism that does the catalysis, down on the electrode surface and make fuel cells that will actually do deeper complete oxidation. So that is another area of interest. Oftentimes, if you think about electrocatalysis, we put a single catalyst down that may do a 2-electron oxidation or a 4-electron oxidation. But here with lactate, if you wanted to use lactate as a fuel, it's a 12-electron oxidation to oxidize lactate to CO2. And although there's no single enzyme that can do that, there's a series of enzymes within pathways that can do that. So there's a lot of work in sort of looking at how we can use what biology does in terms of these enzyme cascades to actually improve performance. And so if you go from 2 electrons to 12 electrons, you can see that you'll get better performance by using a cascade of catalysts that will be able to do this. Sometimes that's complicated. So if you look back here, I have a whole series of enzymes here, lactate dehydrogenase, pyruvate dehydrogenase, citrate synthase, et cetera, to be able to catalyze these reactions. When in reality, we want to be as simple as possible, and we would love to have catalysts that can do multiple steps. And so there's also work at using enzyme engineering, so not synthetic biology. We're not putting it into the microorganism, but we're instead taking enzymes that normally do one particular process, and we're enzyme engineering them to have promiscuity so that they can do multiple steps of a process. fitness. And so this is an example. This is for a glycerol fuel cell. So glycerol is a byproduct of biodiesel production. So it's a high energy density, sort of cheap product. And we can actually use cascades of engineered enzymes. In this case, it's alcohol dehydrogenase, aldehyde dehydrogenase, and oxalate oxidase, where we've actually used enzyme engineering. In one of these cases, directed evolution. In the other, site-directed mutagenesis, to be able to take chemistry take an enzyme that normally has a single substrate and get it to work with multiple substrates so that we can do deep or complete oxidation. Now, I would say over the last 5 years, there's been a lot of focus towards energy harvesting. And obviously, the example that I showed you with wastewater treatment, while they're treating the water with a microbial fuel cell, they're energy harvesting, but there's been a lot more focus on wearable, wearable electronics, wearable point of care technology to be able to sense molecules of interest, so things like glucose or lactate, and be able to actually harvest energy from glucose or lactate that might be in sweat, might be in tears, might be in other biological fluids. So this actually work from Joseph Wang at University of California, San Diego, and he's kind of been the pioneer in terms of looking at flexible technology, wearable technologies. So he's worked a lot with, this is a temporary tattoo that will actually sense, he has temporary tattoos that will sense lactate for fitness while you're working out. He has temporary tattoos that will detect glucose that's coming off of the sweat. He has temporary tattoos to look at toxins that might be present. He actually has it's not a temporary tattoo, but it's actually a sock, so a smart sock that is actually doing metabolite monitoring while you're working out and can tell you different concentrations of metabolites. He has smart gloves, and these smart gloves, so if you're concerned that maybe fentanyl is present or an opioid is present, you can actually swipe it with your glove, and his smart glove actually has sensors built into the glove to be able to tell you whether the toxin is present or not present. So you're seeing a lot of technology that initially was focused on just the sensor, but now focused on, okay, I want my sensor to be wearable, and if I want my sensor to be wearable, I need to think about energy harvesting technology to be able to essentially power the sensors. So you're not easily going to really want to put a bunch of large batteries in your sock or in your gloves, and so the idea that you can essentially have wearables for those applications. And so I'm going to show you one example of that. That's actually an example for a smart contact lens. So they're looking at contact lenses that would have sensors built into them. Those could be interocular pressure sensors for optical diseases. They could also be sensors for sensing things like glucose for a diabetic patient, or they could even be an electronic display. So rather than thinking about Google Glasses and displaying your text message in your glasses, you could think about Google Contacts and sort of having electronic displays in the eyeball. Not my thing, but essentially, you may want to harvest energy for any number of different purposes. And when you start to think about this, you have to think about the fact that now you have a materials issue. So usually we think about making a fuel cell. We're not so focused on making a fuel cell that is flexible. We really have to go back and think about what our electrode materials are. If they need to be flexible, the electrode needs to adhere to the contact lens. The reactant has to get through, so the glucose or the lactate, the electrode has to be conductive, it has to be biocompatible, has to have high surface area so you can get high current densities, and so you end up with a different set of materials design concerns when we start to think about wearables and we start to think about energy harvesting. And so this is just one example. This is from Chuck Henry's group at Colorado State, who's been working a lot with trying to make these sort of flexible electrode materials that will have those properties. So he's adding a high surface area material into the polymer matrix. That high surface area material, so it's a high surface area graphite with polydimethyl siloxane, which is obviously flexible and a similar material to what you would see in the contact lens. And it basically will make a material that is both biocompatible, has good conductivity, has good surface area, and we can actually energy harvest the lactate that is in tears. There's actually more lactate in tears than glucose, but the lactate in tears to be able to generate power. The amount of power that we're producing is enough for powering sensors. It's not enough for displaying your text messages in your eyeball, but it's a step in that direction. If you think about energy harvesting, we can harvest from chemicals, but we can also harvest from solar energy. So this is some work by Dave Klippel at Vanderbilt University, where he's basically been using biological entities. So he's been using the photosystems, isolating photosystems, purifying photosystems, and actually using photosystems for sort of solar energy harvesting. So we don't just have to harvest chemical energy to electrical energy. We can harvest solar energy to electrical energy with bioelectrochemistry. And then finally, my current sort of research focus is whether or not we can use biocatalysis, combined biocatalysis and electrosynthesis to be able to use biology to synthesize interesting molecules. And so we've done this with that nitrogenase that I talked about, that protein that is capable of doing nitrogen reduction to ammonia, and being able to show that indeed the same chemistry that happens in a living organism when we remove the enzyme, put it down on an electrode surface, we can actually get it to function. This is a mediator, methylviologen, so we're mediating the electrons, not doing direct electron transfer, but we can get that nitrogenase to communicate with the electrode. If we take our nitrogenase cathode, we combine it with a hydrogenase anode, we're now doing somewhat like Haber-Bosch chemistry, totally not Haber-Bosch chemistry, but from a reaction perspective, we're taking hydrogen gas and nitrogen gas and making ammonia, which is what we would normally think about Haber-Bosch chemistry, but we just don't have the same catalyst, and we're at sort of ambient pressure and ambient temperature. So we can actually then sort of combine this nitrogenase cathode with this hydrogenase anode to actually make a fuel cell. Fuel cell has a small open circuit potential, but it can produce ammonia directly. And so that's interesting. If you want to think about the techno-economics that Tom talked about yesterday, this is not going to meet those techno-economics, but we don't really necessarily want to make ammonia from an electrosynthesis perspective. Ammonia is really kind of a precursor to what we want to synthesize. And so we've been looking at a number of electrosynthesis systems that would basically be feeding in ammonia, but ammonia is your precursor to a value-added product. And one of those is chiral amine synthesis. So if you look at a lot of drugs are made of chiral amines. They need to be made with a good selectivity. The pharmaceutical industry has been looking at transaminase, sort of using biocatalysis as a way to get that selectivity. The problem is this reaction is actually shifted to reactants instead of shifted to products. So if you think about this from a reactor perspective, you typically would like to be shifted towards your products instead of shifted towards your reactants to be able to produce a lot of your selective product. And so the really nice thing about electrochemistry is we can drive things far from equilibrium. And so we can use ammonia and cofactor regeneration as a way to sort of push this electrosynthesis. So we're using the nitrogenase, but not to make ammonia as a final product, but as an intermediate that is then going to feed into a catalytic cascade for transaminase to basically force this reaction from being reactant favored to being product favored. And this actually works quite well. It's one of the things that bioelectrochemistry can do extremely well, is to take biochemistry that might have the selectivity that you want, and instead of having to do cofactor regeneration in an expensive chemical or biochemical way, we do cofactor regeneration electrochemically and some reactant generation electrochemically to be able to force us in the direction of products. And so we've done this. We've taken a series of prochiral ketones and actually formed selective chiral amines from them. So we're taking that step of making the ammonia, but then using the ammonia for something else. And it turns out that some of these molecules are a lot easier for us to detect than the ammonia itself. There's a pro and a con to that, but definitely, we can think about detecting these with isotopic labels a lot easier than detecting the ammonia with isotope labels. So I was supposed to talk about future challenges. We really still struggle after over 100 years of bioelectrochemistry to really understand the electron transfer pathways between microbes and electrodes and enzymes and electrodes. So right now it's kind of a trial and error process, and we need to get to the point that we have in situ and operando techniques that allow us to take it from being this sort of trial and error process into really being able to understand how those electron transfer pathways work. We need materials to promote electron transfer. So you're only going to be as good as the materials that you have that can promote the chemistry that you want to promote. So we still have a lot of material science to do. Synthetic biology and enzyme engineering are really critical for us, whether we're doing biosynthesis, whether we're energy harvesting, to go to sort of next generation issues. When we think about devices, we have to think about stability. Those microbial fuel cells are stable for 5 years without any problem. Those enzymatic fuel cells are stable for days, maybe months. And so we have to look at shelf life versus operational stability of electrodes as we produce them. We're going to need a lot of work in device engineering. So a lot of the bioelectrochemistry are sort of not well-designed reactors, not well-designed electrochemical cells. And so we're going to have to do some device engineering as we're combining our material science and our synthetic biology together. And finally, we need to integrate into other systems. And this is an example from Plamen Atanasov and Michael Cooney. So Plamen at the University of California, Irvine, and Michael at the University of Hawaii, to basically start combining these sensor networks with microbial fuel cells that can actually be bioreactors to do wastewater treatment, and basically have a smart network system that is sort of combining all of that bioelectrochemistry together. So with that, I would like to thank the funding agencies for the work of my own that I presented, and my group who actually did the work, as well as I presented little snippets from other sort of bioelectrochemistry groups, and I thank them for their work and their well-written papers that I could present to you. So.
Yes. Is there a microphone?
So, Shelley, it sounds as though a biofilm is an artificial organelle, if you really want to think of it that way, in terms of putting the components in you need to do the biochemistry and electrochemistry you need to do. So I would add another bullet to the future challenge, is to get smarter about thinking of creating artificial organelles.
Yeah, I think that's definitely something that there are a few people who are sort of exploring organelles and then artificial organelles. But I think there's this sort of huge area of people in the biomedical sciences who are trying to figure out how to keep biofilms from forming, right? From how to make your antibiotics work as well as they can. And so it's really interesting because they're really focused on how to keep the biofilm to form. But I think we have a lot of lessons to learn from them about how we could promote the biofilm to form, which is really what we want to do in this particular case. So, yes.
Shelley, it was really awesome. I think of this as kind of two regimes in current densities. The kind of microamps per centimeter squared, the sensors and the things you talked about, which I think still have a lot of science, but are really right there. With respect to thinking about this for synthesis, it seems like we're orders of magnitude off on current density. And I wonder there's this fascinating concept of we can abiotically generate these reductants much better than I think plants can and organisms can, but we don't have this, but biologically we have all these selectivity aspects that you talked about. And I'm just wondering what the limits are. I mean, there's kind of an impedance mismatch here of even just simple, solar fuels that run at milliamps per centimeter squared and then electrolyzers run at, hundreds of milliamps and amps per centimeter squared. Where's the limits on kind of space-time yield, how you can really push the titers, if you think of the kind of bioreactor, and then you think of the electrolyzer, how to match those things, and is it possible to push into current densities that would make sense from a productivity standpoint, do you think?
So I guess part of the answer to that is that you have to think about it from either the enzyme side or the micro side. So there are examples of enzymatic fuel cells in the literature with current densities on the order of 25 milliamps per square centimeter at room temperature. That's probably kind of at the upper limit. There are examples of people at 100 milliamps per square centimeter. That's mostly at kind of elevated temperatures. You have to think about what that elevated temperature means. So we work with a lot of enzymes that are thermophiles, and then you can work at some elevated temperatures, so you have some advantages there. But for the most part, in most of these energy harvesting systems, the focus hasn't really been on trying to get the highest current density out. So you're not designing a cell that's really well designed to get current density. So if you look at my contact lens, my anode and my cathode are pretty far apart. So they're just really not, they're designed for the application, not designed for getting high current density and high power density data. As we start to think about electrosynthesis, now we sort of have to change gears, and now it's about a completely different application, and we start to have to think about reactor design. So everything I showed you in terms of electrosynthesis was being done in an H cell. Anode and cathode are very far apart. There's no flow, so you really have to think about those designs, but you also have to think about the enzyme engineering to make them stable in an environment that would, give you higher current densities. So be able to heat it up or even pressurize it.
I'm just wondering, do you know of any interesting research going on at the nexus of all of these studies of the microbiome and how you might integrate some of those bacteria into a possible microbial fuel cell or some sort of electrochemical process?
Yeah, so about a decade ago, there was actually a program in the EU, so one of these EU collaborative programs that was trying to design an energy harvesting biofuel cell for the gut. And that program, I think, probably made the mistake of being entirely focused on enzymes, and I think that's what the EU wanted at the time. It's much more plausible to think about doing that from a microbial system where you're actually going to use the gut's bacteria to generate the biofilm. But that hasn't evolved from the sort of enzymatic side to the microbial side yet.
Hi, Shelley. Good morning. I learned a lot from your talk. If this question is off topic, we can just talk about it some other time. I heard a lecture recently about the idea of implanting nanoscale meshes in human brains to detect electrical impulses spatially and temporally all the time while thoughts are occurring in vivo. Is that something that you have eyes into and that your research relates to?
So my research doesn't directly relate to that, but there are a lot of people who are looking at both sensing electrodes, implantable sensing electrodes for the brain, as well as stimulation. The problem right now is that none of those are stable as long as we want them to be stable. And that's not necessarily a materials issue from the perspective of the initial electrode material, but the body essentially attacking whatever it is that we want to implant. So if you start to think about implantations, whether those are sensors or for stimulation, you're going to have a lot of issues, sort of thinking about how the body is going to respond to those materials.
Great. I think that's one little question I don't have a microphone. So you talked about a lot of cascade, and those are very interesting systems, right? Microbial and enzymatic. How about organometallic and inorganic and enzymatic?
Yes, hybrid systems. So we work quite a bit with hybrid systems. Plamen Atanasov is in the room, so we've had a long collaboration with him in terms of sort of traditional nanoparticle electrocatalyst in combination with enzymes. And then Matt Sigmund, who's also was in the room, in terms of taking organic catalyst and combining organic catalyst with biological catalyst. You have to think about the fact that most of the time your inorganic catalysts are at extreme pHs, so they're extremely acidic, extremely basic. A lot of the catalysis is done in organic solvent, so you have to think about enzymes that would tolerate that, or catalysts that could behave in sort of more neutral pHs or more aqueous environments.
Thank you very much.